So let's move on to an overview of lipids and lipid membranes. So in this section, we're pretty much just looking at the structure of a lipid, basically why they form the shapes they do, and different ways that we can model a lipid membrane or begin to model a lipid membrane. So first of all, let's tackle the structure of a lipid. Just from looking at the structure, we have a phospholipid head, which is hydrophilic, basically water-loving. Then you have a hydrophobic tail. So these are long chains of hydrocarbons, and these are going to be hydrophobic or water-fearing. So we can break it down into a polar head and a non-polar set of tails for a phospholipid. And so here there's a bend, which comes from a double bond. And just from looking at the tail itself, what can we predict about the size and shape of lipids? So you don't just have to have one kink, you can have multiple kinks, which is going to change the shape of your lipid. But the more kinks you have, it's going to spread your tails farther apart. So the farther apart your tails are spread, what happens to your lipid in that case? If you have a whole bunch of lipids that are lined up in one row and all of their tails are spread very far apart, it means that it's going to expand the monolayer that you have. So that means you're going to have wider tails, and if you have wider tails, you're going to have shorter lipids. So when you're forming layers of lipids, the height of each layer of lipid can actually tell you whether the tail is going to be short or if the tail is highly kinked. So those are the only two possibilities. And what I find really great about these is if you're doing surface experiments with lipid monolayers, for example, if different types of proteins or drugs or molecules that you're trying to test out insert into a lipid and change the structure of the tails or incur more kinks, then you'll be able to notice that through AFM imaging or atomic force microscopy imaging, where you'll actually be able to measure the height of different layers of lipids. And then you can see if introducing some element will cause your monolayers or your lipids to flatten or expand. So we've pretty much touched on this already at the very beginning of the term, but what happens when we add oil to water? Basically, what happens when we add lipids to an aqueous solution? So we finally have reached lipids, which are oily things. Now we know that water is going to wrap itself around the lipid molecules so that it can become more ordered and continue to increase entropy for its individual water molecules. So we know water is going to wrap around the lipid molecules or hydrophobic molecules. So what does that mean for our lipids? It means that our lipids, because they're essentially amphiphiles and there are polar heads and nonpolar tails, your phospholipids are going to want to be arranged in such a way that the polar heads will face outwards towards the water or aqueous substance. So if you actually have basically a whole bunch of lipid molecules inside water, this is going to lead us to the creation of a lipid membrane. First, you have layers of your lipid molecules, but they want to get away from water, especially the tails. So in most cases, for the lipid membrane, it's going to be made up of two monolayers of lipids, where you have your tails trapped inside, protected from water because they don't like water, and then your polar heads exposed to the exterior surface because they don't mind water being there. So you probably learned all about this before, but from bio or biochem courses, so this is just a really quick overview. We have your lipid molecules. Lipid bilayers are usually 5 nanometers in height. So when you actually do experiments, you can use that as as a known parameter and see if your results or your experiment was done correctly. If you get 1 nanometer for your lipid bilayer, then it probably means that either you're just imaging a monolayer, or for example, maybe something happened and you're just looking at something else. So there are values that we can use for comparison purposes to see if we've done the right thing. So those would be our controls. Apart from that, you notice all these green blobs sticking in and out of the lipid membrane, and these are proteins. So like I said, we're not going to be going through lipid-protein interactions in this course, but if you're interested in those, I'd be more than happy to talk about it. So when proteins interact with lipids, there can be various different ways it can do so. Number one, it can interact with just the surface of the lipid bilayer. So these are called surface proteins. So they just come towards the surface and they attach attach on the surface of the bilayer. So it can form connections going through the bilayer to the hydrophobic core so that it can be anchored. You can also have transmembrane proteins that go straight through the entire bilayer. These are really common for ion transport molecules, or specifically ion channels. So we're going to go through one example in a bit of these ion channels, which I find really cool. So from the diagram I showed before, which is very similar to this, you have your lipid bilayer. So one layer at the top with the tails facing down, one layer at the bottom with the tails facing up, so it's protected from the blue water. So the one question I have is, can it stay flat forever? Well, number one, I'll say yes, it can stay flat, but only if we create a situation where it's in a preferable and favorable orientation. So if we can anchor the membrane, for example, if we can anchor the bottom polar heads to a substrate, if you have a polar or hydrophilic head at the bottom, that means your substrate will also have to be hydrophilic. So when you're doing experiments, if you actually want to have your polar heads facing up for a monolayer specifically, then you would want to have a hydrophobic substrate so that your tails are going to be anchored and then your polar heads are going to be exposed. So these are just some different tricks that we can use to try and image or analyze the surface that we want to. So back to the question of whether it can stay flat. Number one, yes it can, if we provide favorable conditions. Number one, if it's anchored properly. Number two, you can also use an entire porous membrane, for example. So you can use a gold substrate and basically have a whole bunch of holes in that substrate, and then put one solid bilayer on top of it. And that bilayer can fall apart at the edges. It doesn't matter what happens, but at least for one of the holes that are created, you can, or one of the pores that are created, you'll have a guaranteed flat bilayer going across, and you'll be able to do experiments like ion channel experiments by having water underneath the pore and water above the pore with different concentrations of ions in each. So some examples of neat ways to try and keep a membrane flat for a localized area. So now comes the most important question: can a membrane stay flat naturally? And naturally, no, it definitely cannot, because there's edge effects. Basically, your hydrophobic core does not want to be exposed to water. So that means if you just have a bilayer floating around in water, it's going to want to fold in on itself and create a lipid vesicle. So this is something that I'll focus on in a bit. So here in the next slide. When we take a look at these vesicles, how are they even useful? So in experiments, what you can actually do is, before you even form this, you can put all of your lipids inside a solution with fluorescent dye. So anytime you're creating these vesicles, it's going to create vesicles that have fluorescent dye trapped inside. Now you can take all of those vesicles out, put them into another beaker, for example, and that beaker just has water. So therefore you have vesicles that only contain fluorescent dye in the aqueous solution inside, and you have water on the outside. So you can do quite a few fluorescent spectroscopy experiments with this too, to see if, for example, if this was a model of a bacterial membrane and you're trying to test a drug's effects, you can see if the drug is going to cause ion leakage, therefore killing the cell, by introducing the drug to the solution or your cuvette, and then seeing if your fluorescent signal intensity increases throughout time. So just some examples of what you can do with vesicles, which is very fun. Now, I kind of want to take a quick snippet of this and zoom in so that we can focus on just the bilayer itself. So we've talked about what happens to the bilayer as a whole, but what can happen on the molecular scale? So my question is, what can these lipids do when they're within the bilayer? So what can individual lipids do if they're just there? So number one, the lipids can move across. The lipid membrane is a fluid membrane, so it's always moving. It's got components that move all the time and rearrange themselves. Lipid molecules can rearrange themselves by moving and diffusing laterally. Number two, the lipids can kind of tilt in this way, called flexion. So you have such a way that it can move itself to accommodate for any different types of, for example, transmembrane proteins or different molecules or anchors that are stuck inside. So there's the ability to be angled. There's also the ability for each individual lipid molecule to rotate. So that means if there's a particular kink that's not as favorable, it can rotate so that maybe something can insert into the membrane more easily, or it'll have better organization overall. Next, we have lipid flip-flop. So this is something that I really like. It's super cool. If you haven't heard of flip-flop, it's actually very interesting, and a lot of my PhD was focused on this. So although it's really rare, flip-flop is important when you want to form transmembrane components. So what can these things be? Probably ion channels. So let's say you have a couple of molecules that attach to these polar heads. So they've attached at the top, and that's it. But at some point, maybe these two lipids that are attached to those flip over to the other side. That means you might have some dimers, trimers, or tetramers that are at the top, and some that have flip-flopped to the bottom. And then when these two come together, because the whole membrane is fluid, they're going to connect together and form an ion channel. So instead of having a dimer on top and a dimer on the bottom, they come together and form a tetramer. So once the flip-flop occurs, there's a lot of interactions that can take place afterwards for complexes that are created. So if I were to ask you what are the benefits of a lipid flip-flop, even though it's rare, it's important to allow for a method of transport across membranes and to help promote interactions within the core. So I want to take a look at an example here, and this is basically from my PhD. It's a figure I drew for my PhD for a drug called daptomycin. Essentially, daptomycin, or DAP for short, is a really powerful antimicrobial peptide that is extremely efficient against gram-positive pathogens. So even better, it's good against resistant strains of bacteria like MRSA and VRE. But it works in a very unique way. First of all, it's actually activated by calcium ions, so it becomes positively charged and becomes a cation. And because bacterial membranes are usually negatively charged, daptomycin is activated and becomes positively charged, being attracted to the bacterial membrane. Once it's attracted to the bacterial membrane, it likes grouping together and forming aggregates of 4, so you end up with a tetramer. But sometimes these tetramers are attached to lipids, and all those lipids actually flip-flop to the other side of the membrane. So you have some on top, some on bottom, and the tails for daptomycin, as they come closer and closer together because the membrane is very fluid, they're going to start getting closer and actually sinking in to the bilayer so that they form an octamer. And this octamer actually forms an ion channel, which causes ion leakage, therefore killing the bacteria through membrane depolarization. So here in this diagram, the 4 steps explain how lipid flip-flop occurs. And I just find that super cool because being able to form these different complexes and actually being able to design drugs or antimicrobial peptides that can do such things, to me it's very interesting. And just changing one of the amino acids will stop it from working completely. So I want to take a look at what else can be stuck into the bilayer. So we've taken a look at just flip-flop and how that's beneficial. But for the bilayer itself, you have different, for example, carbohydrates that can be stuck in here. This is cholesterol. That's the green little squiggly line inside each of these. Cholesterol is very important, actually, and it would be good to know that cholesterol helps to promote fluidity of a membrane. And I might ask why it helps promote the fluidity of a membrane. And keep in mind, remember, if you have more kinks, your lipid tails are going to be spread farther apart, which means it expands the lipid membrane, making it more spacious. If you add cholesterol, it's going to cause a little bit of a rift or an additional space between your lipids here, therefore creating more space and more flexibility in movement. So therefore, cholesterol will promote the fluidity of a membrane. And for any membrane that you look at, usually there would be 5 to 30% cholesterol max. Anything more than 30% cholesterol and your lipids will actually start to clump together. So anything in excess, there's always a threshold. Anything more, you hit some sort of maximum concentration and things won't work as they're supposed to. So then what is in a lipid membrane? So there can be so many different things in a lipid membrane. Number 1, you can have enzymes stuck inside. You can have receptors that are stuck there. You can have anchors for these cytoskeletal filaments. So this is something that we won't really focus on in this course. And then you have these transporters and channels, which we did take a look at. Specifically, what kind of proteins can be found in lipids? These are different types of transmembrane proteins. So these are just different examples. You have integral membrane proteins, which is this entire area here, which basically explains proteins that actually insert into the membrane. However far, it doesn't matter. Ones that go straight through are transmembrane. Ones that go through one layer only are called monolayer associated. Ones that are linked to lipids at the top, kind of here, are just called lipid linked. Then you have these proteins at the ends over here when it's when you're looking at these proteins, it might be a protein attached to a transmembrane protein. So then those proteins on the outside are called protein attached because they're not really attaching to the membrane itself, it's to the protein. The last thing I want to talk about in terms of these membrane proteins is that whenever you have a transmembrane protein or any membrane protein in a lipid bilayer, you can actually solubilize them by adding a mild detergent. So the mild detergent, such as Triton X100, those detergent molecules are going to disrupt the bilayer and bring the proteins into the solution. So basically, they take away the lipids from the protein and you get back your protein for reuse if you want.